So I just want to start by thanking very much the members of the committee. It's a really immense honour to be here in the Royal Swedish Academy of Sciences, and especially because we're celebrating this prize to Dolph. So we've just heard that very nice tribute from Katie. He's also had a big effect on my career, both himself and his ideas. So if there's anything worthwhile in what I'm about to say, then it's dedicated to Dolph. I started life as a population ecologist, and I have toddled into genomics rather belatedly. So a lot of the data I'm going to talk about today are genomic data. They're quite recent data, and I'm kind of still in the process of understanding them, so this is a work in progress. There's a couple of points, I guess, that I want to kind of try and make. So first of all, I think in this genomic era where we're being deluged with some really amazing data, we're actually rediscovering things that were perhaps known up to 100 years ago through the work of very careful natural history and very careful classical genetics. So that's kind of the first thing that you'll see as a theme through my talk. The other thing is that there has been a lot of interest in this idea of parallelism. I'm going to talk a bit on the next slide about what that actually is. But I think this idea of persistence that has started to come up recently is really quite an important one. My interpretation of the evolution of the stickleback is kind of different, slightly different from Dolph's and Katie's, because I think there's more evidence, at least in some systems, of a kind of historical component to the evolution of some of these pairs, and perhaps with some of these data, I'll be able to put that idea in your heads. So, parallelism is this phenomenon where you get recurrent similar forms evolving in similar environments in different places. The anolis lizards that have been so well studied by Jonathan Lossos in the Greater Antilles provide a very, very good example of this. So you I have to get used to this pointer, different ecotypes living in different parts of the forest canopy, so on twigs, or between the trunk and the crown, or between the trunk and the ground. And you get those reoccurring across the different islands of the Greater Antilles and appearing to be very similar. And so this kind of pattern is very strong evidence of natural selection reoccurring in similar ways in different places. Now, a number of people have made the point that this kind of pattern can come about in different ways. So often it's assumed, you know, if you have two different ecotypes of something occurring in different lakes, that those are independent evolutions in different places. So this is from Rui Faria's work, so this kind of model where you have this independent evolution in the different places. Then there are a number of different possibilities. So at the other end of the spectrum is that these ecotypes have actually evolved only once, so maybe, let's say, in Lake A in this case, and then have then dispersed and colonized other similar habitats in different areas. And in a way, a kind of halfway house is that these things are reoccurring in different places, but they're using this kind of older adaptive variation that Katie and Dolph have already talked about. And really, it's really hard to separate these different possibilities, so this possibility from this possibility and this possibility, that's especially the case where you have this possibility of this older shared variation, and it's also particularly difficult in situations where you have secondary contact, so where you have sympatry between the ecotypes, where they can be exchanging particularly neutral parts of the genome, and then the picture of their history can be quite easily lost through time. So one of the common kinds of ancestral polymorphism that's already been talked about are these inversions, so where you get a region on a chromosome that has been flipped around through some genome flux event and then becomes effectively protects the loci that are within that region from recombination. If there are loci in that region that predispose to adaptation, then you effectively get a kind of protected section. So what Nick Barton has referred to as little species, so protecting these kind of adaptive cassettes. And there's been quite a lot of recent genomic work finding that these inversions contribute quite major genomic variation in different circumstances. So in the variation between sunflower ecotypes in the United States, in the variation between cod, resident, and migratory ecotypes off the coast of Norway, and then in different ecotypes of deer mice, again, in the US. So in all of these cases, the inversions are contributing quite large, important parts of the adaptive variation that we see. Of course, that's an idea that was known to Dobzhansky and Sturtevant, almost 100 years ago. And so that takes me on to Dobzhansky. He's already featured quite strongly in this symposium. Many of our ideas about speciation go back to him, the ideas about reproductive isolation. So this is quite a well-known figure. And here, Dobzhansky is imagining a species, and the black lines within the species, oh no, if I can get this to work. So the black lines here are imagined to be independent populations. And Dobzhansky imagined a number of different possibilities. So you have a single species, say at A, begins to diverge at B, and becomes two different species at C. Something that Dobzhansky didn't consider was a more reticulate idea of this. So this is from Veron's book about speciation in corals. And Veron in this brings in the idea that these diverging lineages can collapse back together. And so you can perhaps get relatively long-lived lineages that diverge and then come back and rejoin with other lineages. And when you think about it, it's kind of almost necessarily true in circumstances where you have these ancient polymorphic inversions reoccurring, because presumably they have evolved somewhere back in the tree and they're popping up again at later stages. And this also recapitulates an idea that Godfrey Hewitt particularly championed quite a long time ago, so starting sort of 40 years ago. Godfrey Hewitt was very interested in the pattern of genetic diversity across Europe. So a number of different taxa here, grasshoppers, hedgehogs, bears, fish. But Godfrey Hewitt proposed a model in which the ice ages effectively came down and the ice ablated any variation that accrued. And then lineages had refuge here in southern places away from the ice sheets. And when the ice melted, then they spread back. And they might have diverged in the meantime. They come back together. In some places, they form hybrid zones. And in those days, these lineages were normally taken as being marked by mitochondrial genes. So in those days, it was really considered that mitochondrial genes were good neutral markers of population structure. More recently, that idea has come up again in our own history. So in human ancient DNA work, so of course Svante Pääbo won the Nobel Prize for really getting this work going, picked up by people like David Reich. And one of the patterns that comes out of that, certainly within Europe, is that you get these waves of humans that have diverged to some extent, traveled across Europe, and then come back into contact. Sometimes those populations have just mixed. Sometimes it seems that one population has replaced another. But the point is that there's this geographical and historical context. So that brings me on to the stickleback species pairs. We've already heard quite a lot about them. So we have the work on marine and freshwater species pairs by Felicity Jones and David Kingsley's group. Then, of course, all the work that we've heard so much about on benthic and limnetic pairs by Dolph and Katie, and then also stream lake pairs in British Columbia as well. So I'm just going to, the first part of this talk, I suppose, after that introduction, is I'm going to talk a little bit about the benthic limnetic species pairs and about, I suppose, my model of where these might come from. So this is a map from Don McPhail's work in a very famous stickleback book, Bell and Foster, from 1994. So mapping the occurrence, you've already seen a very similar version of this map in Katie's talk and in Dolph's talk. So there were 6 original populations in 4 different watersheds, and it's interesting that when Jenny Gow went looking for further species pairs, something like 20 or 30 years later, the one pair that she found was in fact fitted on the same map. And the maximum distance between any of these populations as the crow flies or the fish swims is 60 kilometers. They cover a total area of 760 square kilometers. So in terms of the total distribution of stickleback across the globe, which is marked in grey here, the occurrence of these species pairs, which are obviously very special, is very, very geographically limited. And Don McPhail was very aware of this when he described the species in the beginning, and he was very aware that any explanation for their existence had to take account of that fact. And the idea that he proposed was this idea that Dolph's already talked about of a double invasion of these lakes from the sea. But one of the things that Don talked about was that suggests some special set of circumstances occurring in this small area. And Don came up with this idea of what's become called a double bounce in the sea level. So he had this idea that sea level started high, as you've seen in Dolph's lovely animation, but fell to a point where the lakes became isolated, then rose again subsequently to allow a second wave of immigration to these lakes. Now, the geomorphological data don't support that idea of a double bounce in sea level. There just seems to have been a decline in sea level following the ice as isostasy happened, and then the sea level has slowly risen again to its present level. So some years ago, I had a project that collected data in British Columbia, having worked with Dolph some years ago, and we collected populations from across this area of southern British Columbia and quantified phenotypic variation, but we also did rad-seq data in those days, so things have moved on. But you can see on this figure the distribution of these populations around the Strait of Georgia, and we included in here a couple of the species pair lakes, so Priest and Paxton. Now, more recently, a postdoc of mine, Laura Dean, has been analysing these data, and one of the patterns that jumps out of them is that within these data, so this is something called a co-ancestry matrix, and it tells us about the relationships between individuals. So it tells us something about the extent of their likely shared history. So as you go from yellow on this side through orange and red up to pink and blue, that's increasing evidence of a shared history. And no other information apart from the genomic variation is put into these analyses, but what comes out is this pattern in which it's very clear the squares here are different populations, and you can see here that we've got the Paxton and Priest benthics clustering together here, and the Paxton and Priest benthics clustering together up here, and then some evidence of shared co-ancestry between them. But the next most obvious pattern is the fact that you seem to have 2 clusters of co-ancestry. So there's a cluster here, which we call cluster 1, and a cluster here that we call cluster 2. That pattern, interestingly, is also what I should have said, sorry, is that these clusters, so they're marked on the side here in blue and red, and there's no particularly obvious geographical pattern in British Columbia in how the fish in these clusters are distributed in space between different lakes. There's a very similar cluster in Alaska, so we collected very similar data in Alaska. There you have rather a similar pattern with the populations and then two broader clusters. In the case of Alaska, the two broader clusters are two different collecting areas. So the Kenai Peninsula south of Anchorage and what's called the Matsu Valley north of Anchorage. There are differences when you look at the phenotypic data between various traits within these clusters. So most of these populations are single species populations, but they show some of the same patterns of traits as the benthic and limnetic ecotype species do in the species pair lake. So there are differences in the number and length of the gill rakers that are an important trophic trait. There are differences in the armor and in the shape. So here on the axis we've got marine fish from Oyster Lagoon and Little Campbell River, both in British Columbia, and then the two clusters. When you plot a population tree for the different populations, the two clusters fall out together, and it appears here. So these are the benthic-like fish in the red here, including the Priest and Paxton benthics, and then we've got limnetic and marine-like fish down here, including the Priest and Paxton limnetics. And this population tree rather suggests that benthics may have had a shared genomic origin or genetic origin at some time in the relatively recent past. And when we go back to the spatial distribution of these populations, I'm slipping slightly into speculation here, but the populations of the benthic-like fish are all distributed rather close to the shore around the edge of the Georgia Strait. So sort of consistent with the distribution of this blue polygon that I've overlain on the map. But the interesting thing about this blue polygon is that it's actually nothing to do with the distribution of the species. It's actually the effluvia, or the evidence of the effluvia from a massive flood that took place out of the Fraser River when one of the ice-dammed lakes drained 11,000 years ago. For 500 cubic kilometers of water poured down the Fraser River and spread out across the Strait of Georgia, taking all sorts of clay and rubble with it, and turned the surface waters of the Strait of Georgia to something like brackish water for a period of probably at least several weeks. So this is a speculation, but it's possible that these benthic populations had a shared origin in some freshwater lake that was up the Fraser Valley and have then been effectively washed out by this flood. Okay, so at that point I'm going to turn to a different set of populations, so more my main interest, and these are populations of stickleback in the North Atlantic. Now, in the Pacific, it appears to be the case that there's a relatively uniform marine population, and what is less known is that that is definitely not the case in the Atlantic. So in the Atlantic, there are actually reoccurring species pairs that live in salt water. So these are photographs from my main study site on the island of North Uist on the west coast of Scotland, and we find co-occurring in numerous different locations fish that look like this. So we know, to most of you these will just look like small fish, but there are actually very many differences between them. Prominent between them is the difference that these fish are resident in these coastal saltwater lagoons all year round, whereas these migratory fish come in from the sea in the springtime, breed, and then the juveniles go back to sea at the age of about 6 weeks and spend 2 years at sea. So there are differences in life history between these fish, differences in size, differences in morphology, differences in mating behavior, differences in parental care. So quite substantial differences between these ecotypes. And it turns out that these pairs occur elsewhere in the Atlantic, so they've been documented by Mark Ravenay occurring on the west coast of Ireland. More recently, myself and Isabel Magalias have found them co-occurring in saltwater on the north coast of of Iceland. And so just to kind of emphasize this point about these fish being relatively reproductively isolated, the migratory fish are completely plated, the resident fish are low plated, so you would expect any hybrids between them to be partially plated because of this dominance of the Eder allele, or partial dominance of the Eder allele. And when we look, well, we looked through 1,500 fish and we found 1.4% among adults of partially plated fish, so rather a low level of hybrids evident in adult fish. I'd been thinking about these fish off and on for about 10 years before I thought to go and look at some of the older literature. So I went back to some of the kind of classic work on stickleback in Europe. So this is a paper by Bertin from 1925, the first really systematic survey of stickleback in Europe. These are data from a fjord in Denmark, caught in the wintertime, and it's probably easier. So he's plotted partially plated, fully plated, and low plated fish. And when you make that more obvious, it's clear that you have small low plated fish, large completely plated fish, and then some intermediates. And Bertin ascribed this to a Lamarckian mechanism. So he thought that they were acquiring plates because they needed them as they grew up. Hoyts picked up the same theme when he looked at stickleback variation in Europe and particularly in Belgium in the 1940s. He found that, so this is a slightly odd figure because this axis is inverted, so he found a set of fish towards the south of Europe that he called A's, and a set of fish towards the north of Europe that he called B's, and this axis down here is also inverted, so these fish tend to be completely plated, and these fish tend to be low plated. He found these different types co-occurring in freshwater and brackish water and saltwater in Belgium, and he did various experiments, found quite large physiological differences between them. Munsing then, 20 years later again, picked up the same idea and the same themes, and he proposed this model of two lineages of stickleback, so one originating somewhere to the north and spreading down into Europe, and another one originating in the south and spreading northwards. Now, the standard model for stickleback ecotype evolution would be this idea of some panmictic oceanic population, the kind of model that Dolph's talked about for the Pacific, where you get evolution of fish adapting to fresh water, diversifying, becoming different ecotypes in different places. But the work by Munsing suggests a different kind of model, something more like the kind of model that Hewitt proposed, where you have different lineages. So in refugia, perhaps on the coast of America and one on the coast of Europe, spreading and meeting around the North Atlantic. And when we started looking at the species pairs in Europe, one of the first most striking things that we found was that indeed these different ecotypes vary quite strongly in the different proportions of mitochondrial haplotypes within them. So the resident fish, both in saltwater and now I'm showing you freshwater resident fish, are largely of a European mitochondrial haplotype, and then the migratory fish are at least on UIST, are a 50-50 mix, pretty close, of a European haplotype and a transatlantic haplotype. So that terminology comes from a paper by Mackin and Marilla from a long time ago, in which they looked at mitochondrial variation across the North Atlantic. They found these two lineages, so they found a European lineage in Europe, and then they found evidence of a transatlantic lineage occurring on the east coast of North America and in the western parts of Europe. And those, again, seem to associate with these differences that we see in these ecotypes. So a student of mine, Megan Barnes, has added to that Mackin and Marilla data set. So we've now got mitochondrial sequence from over 2,000 fish, 250 populations, mostly in Europe because European populations have been much better sampled than the ones on the east coast of North America, at least until relatively recently. And so what we find indeed is that, these axes are showing the proportion of transatlantic fish. So in American samples, these fish are largely of the transatlantic lineage. In European populations, the fish are largely of the European lineage. That gets more interesting if you break this down by habitat type. So in fresh water, where the fish are low-plated or completely plated, the transatlantic lineage is rare anywhere, in Europe or in North America. In saltwater, in contrast, complete fish tend to be of the transatlantic lineage, and low-plated fish tend to be of the European lineage. So this pattern is interesting in the context of a wider phylogeny of mitochondria of stickleback. So this is work I did with Felicity Jones some years ago now, looking at patterns of selection across the mitochondrial tree for three-spined stickleback. So this is from complete mitochondrial genomes, and I use three different methods for trying to look at selection, so TreeSap, MEME, and CodeML. So CodeML is generally kind of taken as the gold standard for this kind of work. The pattern here is interesting because for the TreeSap, you see there are basically two foci of evidence of selection. So in the split between what I'm going to call the kind of older stickleback populations in the Japan Sea and in the kind of Beringian region, between those and the newer populations in the Pacific and in the Atlantic. And then there's also these differences around the split between the different Atlantic lineages. And when you add in the MEME and the CodeML analyses, the really strong evidence of selection is in the Atlantic on the mitochondria. So that makes us immediately wonder whether there is in fact selection going on the mitochondria. It's supposed to be a neutral marker, but perhaps not. You know, obviously the mitochondrial genes are coding for some of the most basic, important physiological functions. And again, this is work by Megan Barnes. So this is looking at microrespirometry. And these are quite early data. These are data that she just put together actually a couple of weeks ago. So, you know, I don't want to put too much stress on these. But in this first pass, it does look as if there's some difference in the respiration. So the x-axis, the y-axis here is the respiration. We've got two different tissues and three different temperatures. And it appears that the European fish are, the fish with the European mitochondrial haplotype are respiring better at low temperatures. And I should emphasize that this is within the migratory fish. So there's no confounding difference, or there's unlikely to be a confounding difference in the nuclear genome here. So this puts into our mind the possibility that there is a kind of energetic difference between these fish. Another one of my students, Henry Lewis, is looking at differences in FADS2 copy number. So that's a gene that is associated with the elongation of fatty acids. And in whole genome data, we find that there's a pattern. So the migratory fish have the lowest copy number variation, the resident fish have intermediate, and the resident fresh have the highest. And this is consistent with this pattern in the ecology, which is that fatty acids are much more available in marine systems than they are in freshwater systems. So freshwater fish are struggling to get the fatty acids that they really need. When he looks at the actual fatty acid content of fish in the wild, he finds the exactly opposite pattern. So resident fish having the lowest despite the highest copy number variation, resident saltwater in between. Migratory fish very variable for reasons I can talk about, but so there seems to be a pattern of counter-gradient variation. This gets interesting when you look at fish that are raised in the aquarium on standardized diet, so they're just being fed bloodworm. And in those cases, the freshwater fish seem to be able to get quite enough DHA, but the migratory fish in the aquarium really struggle to get, or to make enough DHA apparently. And that is consistent with another result that we've had. So I don't unfortunately have the kind of experimental ponds that Dolph has, but we've tried to do some similar things where we've just introduced sets of fish into kind of farmyard ponds in the UK and have then looked at whether they were successful. And we've done that with purely marine fish, and we've done it also with F1 marine by freshwater fish. And we've done it a few times, so normally it's something like 300 fish going into a pond. And when we do that, we find that the probability of the purely migratory fish establishing is significantly lower than the probability of the F1 fish establishing. So this suggests that these migratory fish have a problem in adapting to fresh water. I'm going to go on. The last bit of this talk is then looking a bit at the genetic differences between these two different types. So here we've got, in a location where there seems to be strong reproductive isolation between the forms, we're seeing differences. So FST here is this measure of relative genetic differentiation, and in these pairs of fish co-occurring, the background level of FST is quite low, but we have some really strong peaks. So here and here. So quite similar to some of the data that Katie has shown. And here, coming back to some of the questions that Katie had, the lines here, so here and here and here, it's really quite hard to work this thing, and here, are the inversions that are kind of known about that have been thought to be associated with freshwater, saltwater divergence, especially in the Pacific. This is a speculated inversion from more recent work on chromosome 9. If you plot these FST data, which are a relative measure of genetic divergence, against absolute genetic divergence measured as dxy, then in general there's not much of a pattern, but there is a group of points out here where both the relative genetic divergence and the absolute genetic divergence appears to be quite high. So absolute genetic divergence takes time to build up. And when you look at those, the location of those high DXY and high FST locations, they again map to some extent to these inversions or to regions that are otherwise known to have low recombination. So these are likely to be these ancient regions that are being reused in this circumstance for adaptation. If we zoom in on some of these inversions, then we see some quite interesting patterns. So chromosome 21 here is in the Pacific associated, well, and in the Atlantic actually, is supposed to be associated with adaptation to fresh water. But here we've got two saltwater populations here and here, then a population that's resident in saltwater and a population that's resident in freshwater. And there appears from this first pass to be rather little difference between these different populations for that inversion. The chromosome 11 inversion shows the classic difference between freshwater fish and all fish living in saltwater. But then the interest starts to go up for chromosome 1, where you have saltwater fish having one version of the inversion, and then fish that are resident having mostly the other variation. So it appears that this inversion on chromosome 1 may be more associated with migratory behavior than with adaptation to fresh water. It's interesting that there's a bit in the middle here that's been apparently reinverted, probably a kind of double crossover. And right in the middle of that is a gene that is pretty much known in fish to be involved in osmotic regulation, a gene called ATP1A1. These are, as I say, we've only seen these data in the past week or so. But chromosome 9 is looking really interesting. This is a large inversion on chromosome 9 that's not obvious in the Pacific, but it's interesting because it seems that most fish are heterozygous. So these are from short read data. We have to be a bit careful about that, but other sources of information suggest that this is an inversion that's segregating, and it suggests that there's heterozygous advantage for this inversion across all populations. So we don't know, it's a large block. We haven't looked at the genes that are in there. We do know that there's one associated with sperm motility, but otherwise that's work to be done. So that's about the differences between fish where they are well reproductively isolated, but we also have a hybrid zone on North Uist where the three different forms of the fish meet. So these are stained fish that have been stained for bone. These fish were caught in a single trap in a location on North Uist. And so you get an immense amount of phenotypic variation in this location. It's a very small location, but for some reason the reproductive isolation is breaking down there. And you get fish that look like residents, saltwater residents, you get fish that look like saltwater migratory fish, and you get fish that look like freshwater residents. The points over here, the colored points show the parental populations, if you like, where there's good reproductive isolation, and these are phenotypic data. So we've plotted the variation in the phenotypes between the populations, and then the white points are the places in the hybrid zone where there's a kind of a somewhat of a collapse of the phenotypic differentiation, but really rather less or rather fewer hybrids, clear hybrids in this region than you would imagine in a normal hybrid zone, suggesting that there is still something that's maintaining the differences here. And if you look again at the FST in the hybrid zone between the two different saltwater forms, it's reduced, but some of these inversion and other differences remain. If you look at the hybrids between the fish, then it's interesting that most of the hybridization in this location appears to be going on between the migratory and the resident saltwater fish. There's odd fish that look like F1s or F2s, quite a lot of fish that look like back crosses, and very few fish here. Now, there's various patterns that can produce this, but this does suggest that there is selection against these back crosses into the migratory population. And a bit more data for that comes from, so now we're looking at the genomic data for the hybrids, and you can see again the three clusters, hybrids in between. It's interesting that the saltwater residents that are normally low plated in the hybrid zone are commonly completely plated or partially plated. And so we know that there's gene flow going from the migratory fish into the saltwater resident fish. We know also that some of these fish are partially plated, and we would expect, are either heterozygotes. So we would expect those heterozygotes to be partially plated, but in this circumstance, they are completely plated. And that suggests a kind of model a bit like Dolph's of having to maintain some kind of phenotypic integrity. So these are fish that have been to sea, they've been through the selective filter of migration, and it seems likely to us as a model that they are being weeded out in the saltwater environment, in the open ocean, by very strong selection against fish that don't have a complete set of tools for that environment. So we have this model, a bit like the model that Dolph talked about with the limnetic and benthic, but in this case, the idea here is that these differences are being maintained by this differences in the tendency to migrate. So you can be a resident fish with this set of phenotypic characteristics, or you can be a migratory fish with this set of characteristics, but falling in between those two stools is kind of game over, very strong selection against them. So this may be a mechanism that's maintaining these differences, and indeed, there's growing evidence actually that differences in migration can maintain differences between ecotypes within animal populations. So various fish species, various other vertebrates, so caribou and orcas. And then also in birds, that you get genomic variation between closely associated migratory and non-migratory populations, or populations with different migratory routes. And so in terms of going back to Rui Faria's work, this suggests to us a situation, the mitochondrial data suggests that within the Atlantic we've got a single origin of ecotypes on either side of the Atlantic, but there is reuse of these shared ancestral populations, so it's something between these two, and a model that perhaps looks more like this, more like the kind of Godfrey-Hewitt model. And so just to finish, really, I just want to say, so parallelism is a very important, interesting phenomenon. It may have quite complex origins. This idea of persistence may be important where you have secondary contact of older lineages. I think we don't know too much about those mechanisms of persistence. It's probably some combination of selection plus genomic tricks like inversions, which are still driven by selection. And it appears that migration may really be quite a major selective agent. So I just want to close by, thanking especially some really amazing young scientists. I don't do any of this, I just talk about it. So some amazing young scientists I've worked with recently, or I'm still working with, and my fantastic long-term technician, Anne Lowe, and then funding, especially from the Natural Environment Research Council in the UK.